helping you to improve your work from proposal, also chapters, and generally your thesis, and improve that presentation. So I just would like to really make sure that this time you enjoy, you ask questions, you are kindly critical towards the others, and yes, welcome to this annual, what has become an annual event for SAIAB. It's very important. I would like to thank the organizers. There is a big team that has worked with non-stop in the last, I don't know how many months. So thank you, Gwyneth and her team. And also I would like to thank, I know that at 12 o'clock we're gonna have, we're gonna show support to this shutdown movement that is happening today. Well, I'd like to thank the people that made you have this ribbons to celebrate, or rather to celebrate, to remind ourselves throughout the day about this important event. These are Aku and Ave. They are the daughters of one of our PhD students. So everyone really was literally involved in this event. So just the last note, welcome everyone, and I hope you enjoy and you get inspired by today. Thank you.
Thanks, Francesca, for that lovely opening message. And without further ado, I think we're going to just dive right in. So our first keynote speaker is Prof. Rajakaran, Anusha. Thank you very much for coming all the way from Cape Town to join us. A little bit of a background. So Prof. Rajakaran is an estuarine ecologist specializing in submerged and emergent macrophytes, including mangrove forests, salt marshes, and seagrass ecosystems. She got her PhD in botany, just shows the cross-disciplinary function of SAIAB, from NMU, Eastern Cape, and were in 2011 and was appointed lecturer in the Department of Botany at Rhodes University, and then later moved to UWC, where she is currently the head of department. Her research group works across estuaries nationwide. Along the East Coast, the team focuses mostly on heavy metals, microplastics, and structural dynamics of mangroves. And on the south, they look at the fauna, utilizing different habitats to better understand and prioritize nursery areas of fish species. On the west coast, their work examines the resilience of salt marsh ecosystems to disturbance and quantifies carbon stocks within the Berg River estuary. And then within the Cape Metropole and across South Africa, they do microplastics research to assess the occurrence of these pollutants across habitats and food webs. Prof. Rajakaran also serves as the chair of the Western Indian Ocean Mangrove Networks Policy and Advisory Group, where she contributed to the development of the Regional Mangrove Action Plan. And she's also a member of the Global Wetlands Group, which works to translate scientific understanding of wetland health and into accessible information for broader audiences. So without further ado, I'd like to welcome Prof. Rajakaran. Let's give her a wonderful hand.
Good morning, everyone, and thank you for this lovely invitation. It's been exactly 10 years since I left Rhodes University for the University of the Western Cape. So it's really nice to be able to come back and to give you a little bit of overview of what we've been doing. And it's really nice to see some ex-UWC students in the room. It just shows how important it is for our students to travel across the country so that they can learn from each other. So I'd like to talk to you about aquatic research in the Anthropocene and what estuaries teach us about being resilient. And before we do that, as a lecturer, we always like to get the crowd engaged. So I'd like you to think about some of the threats that you are most concerned about when it comes to our estuarine or aquatic systems. So maybe write down just your top 3 threats that you think are the most important or the most critical to your work. Well, not yet. That's why I became a botanist, because only animals bite. Okay, and then if you've done that, I want you to shout out, what are some of your favorite estuarine or aquatic systems? Where's the place that you would want to go back to and make sure is kept in a pristine state and is lovely for everyone to enjoy? Just shout it out. Kawi. Nice, huh? Yeah, Ngazana, great. Okay, so all of those are really great examples of places that we enjoy and that we get some kind of comfort from and lots of services. So if we go back to those negative threats, do you see some of your threats that you wrote down? Okay, so I think we have a very good understanding of the different threats. We may not have a complete understanding of how they affect our aquatic habitats, and from my perspective, especially the plant habitats, but we have a good idea of what we need to be dealing with. And of course, there's an ocean emergency. We all know this. If we think about coastal eutrophication, the very first slide I showed was of Isipingo estuary, and no one shouted out Isipingo. Did anybody? Because when you go to Isipingo, the first thing that hits you is the smell. It's an incredibly chemical smell that almost burns the lining of your nose, and you don't really want to be around that. And you saw the water hyacinth with the mangroves. That was the very first time I've seen so much water hyacinth just adjacent to the mangroves. That shows the amount of nutrients that are coming in there. So Isipingo may not be a very attractive estuary, but for mangroves within KwaZulu-Natal, it could be quite an important one. So we shouldn't forget it. If we think of ocean acidification, ocean warming, plastic pollution, and overfishing, I think when we look at SAIAB and SAIAB's profile, you're dealing with all of these really important issues, and you are contributing really meaningfully to the ocean emergency. And these pictures and these articles are very much forefront within South Africa's news headlines. Are we treating our estuaries an extension of our toilets? Are they the toilets of South Africa? This is where all the nutrients are going into. How does that have an impact on the different fauna and flora? And is there resilience to that? Are there alternative states, or are there tipping points that we are now pushing into? So of course, our estuaries connect the land to the sea. I don't need to tell this audience that. So we have this continuum of what's happening in the terrestrial environment affecting our rivers, and what's happening within our rivers affecting our estuaries. And of course, our estuaries are also influenced by what comes in from the marine side. So they are really this melting pot of different anthropogenic activities. Especially within the Cape Town region, we know of many sewage spills within KwaZulu-Natal at the Umhlanga estuary. I don't think anyone screamed out Umhlanga. There was that massive chemical fire during the riots. All of that water from when the fire department was trying to put out the fire ran into the Umhlanga estuary, led to massive fish kills, massive levels of loss of plant habitats. And we've been trying to see, is there recovery from that? What can we learn from those kinds of events? And of course, these flows into our estuaries changes the hydrology, it changes the nutrient dynamics. Recently, I think it was the mayor of Cape Town talked about the Dip estuary. And the Dip estuary is another one of those estuaries when you go there, the first thing that hits you is the smell. And then you see the methane bubbling in the water. And that's quite a sight. And if you pull a seine net, you get sludge. You get nothing else. And he was talking about how they were going to go into the Dip River and the Dip estuary and try and see if they can clean that up. So there's a lot of impetus and movement as our data, all of our data gets pushed into the mainstream. And that's really important for us to do. So of course, I look at the biological response, and especially from a botanical point of view. So during this particular presentation, I want to focus on the positives. We all know what the negatives are. We don't need any more negativity as we get towards the end of the year. So there will be some negative, but we want to see on what is that resilience. And of course, what is resilience? It's the ability to absorb disturbances. If we look at ourselves, what determines your resilience to disturbances into your life? Is it your network, your support system? And our habitats, our mangroves, seagrasses, and salt marshes have particular traits that allow them to be resilient up to a particular point. And biodiversity plays a key role in that. So our keystone species need to be present in order for that resilience to be present. The connectivity, the hydrology, and the sedimentology, what's happening with the flow of fresh water, what's the quality and the quantity, how much sediment is being moved into the estuary, and is that actually a problem or not? And we'll look at an example of that. The habitat regeneration potential, the role of the fauna, and then of course those ecosystem services which we are heavily dependent on. So can we look at how those are changing over time, and can we redeem them? So I'll start with mangroves because that's where I started as a student. So with my training, I did a lot of mangrove work from my honors to my PhD, and I've continued with mangroves. So when Gwyneth mentioned the Global Wetland Foundation or team, and what they're trying to do is to look at a health assessment for different kinds of blue carbon ecosystems. So if you go to the doctor, what are some of the indicators that a doctor would use to assess your health? Blood pressure, that's probably one of the first things. Sugar, your temperature. Do you have a temperature on that day? If they're looking a little bit deeper, they might go for your weight. They might go for your height. They might try to do some correlations between that, and that always freaks me out. Or they might look deeper and start to look at your x-rays, okay? So there's different ways to assess the health of a human being. How do we do that for a mangrove forest? If I walk into a mangrove forest, what are the first things that I'm going to look at to determine if that is what we'd call a healthy mangrove forest? And I think globally, the main thing we try to look at is mangrove cover. So has the amount of, or the hectares of mangroves changed over time? And that can be quite easy in the countries where there's thousands of hectares of mangroves present, or hundreds of thousands of hectares. And it can be a little bit more difficult for countries like ourselves, where we have less than, I think, less than 3,000 hectares of mangroves. So mangrove cover can be a very good indicator of something happening. But when we look underneath the canopy, so we can look, you can use GIS to look at what's at the top, but it doesn't tell us what's happening underneath. And if you walk into a mangrove forest and you don't see any seedlings, what does that mean for the forest? If you don't see any, if you see seedlings but no saplings, what does that mean for that mangrove forest? If the trees are present but they're not producing any propagules, how does that affect the longevity of that particular system? We can also look at carbon stock. So I've talked about blue carbon habitats, and have you all heard that term? Yep, it's a very topical term. We want to be able to assess the carbon stocks within our mangroves, seagrasses, salt marshes. We kind of forget the reeds and sedges, but the reeds and sedges are also quite important. And is that changing over time because of anthropogenic impacts? The sediment is critical. The sediment has a huge impact, not just on the trees themselves, but also on the different types of biota that are living in them. So it affects the macrofaunal composition, your fish composition, your bird composition, and your rare species. Are those rare species there? In our country, if we go up to Corsi Bay, we will find the mangrove whelk, which is not the snail that's on the tree, but the actual mangrove whelk, which is quite a big mollusk. We sometimes find it at Durban Bay as well, and Durban Bay is within a working harbor. Are those characteristics influencing the presence or absence of that mangrove whelk? So these fiddler crabs, and we have a very excellent mangrove crab person in our country, Nazreen Peer. And we work quite well with her to understand the role of biodiversity, and especially of our crabs, and what are the drivers of change for their biodiversity. So we know our crabs are keystone species. They are ecosystem engineers. They allow the sediment to be oxygenated by their burrowing. They cycle our organic matter. They influence nutrient dynamics as well as water movement. So if we lose these functional groups, how does that affect the overall functioning of our mangrove forests? So if we look at the resilience of mangroves, we can look at a few examples. So if we look at mouth closure, in the past, there's been quite a few examples where an estuary mouth will close, the water level rises, and that leads to a mass mortality. So we have a disturbance. What's the capacity of that particular ecosystem to bounce back from that disturbance? And we looked at an estuary called Kobonqaba, which is in the Eastern Cape, and we looked at the recovery. So we lost almost 90% of the mangrove trees. And the very first time we walked in there, and that was about 10 years ago, it looked like a mangrove graveyard because it was just the actual trees, no leaves, and those trees didn't decompose very quickly. So for years and years and years, you had these structures present. Recovery was in stages. First, the sediment had to stabilize, and you had a very high level of microphytobenthos. We then had salt marsh coming in because the seeds were sitting in the sediment, and therefore you had germination of your salt marsh. And with that salt marsh, we often have a nurse plant function where your salt marshes allow your mangroves to establish in those areas. And then it's about the adult trees that are actually present and the propagules that can come in from other estuaries. So all of those added to the story of recovery after that major disturbance. So you had these different states over time, and it's probably taken a good 5 years to get a growing mangrove forest back into that system. If we look at the extreme flooding, do you remember the extreme flooding that happened about 4 years ago now? And that happened in the Durban area. So we went back, we had some data from 2018 before the flooding, and we went back after the flooding to look at what was that impact of that extreme flooding event in the Durban area. And the trends were very similar before and after. We didn't walk into mangrove forests where there were dead trees present, where scouring had happened and trees had fallen down. We walked in when we still had our adult population quite stable and very much present. What we've noticed in our KZN estuaries, is the fact that you can have very tall trees and you can have your seedlings, but you have no saplings. So the trees are producing propagules, and of course mangroves have that very helpful property where the seeds will germinate on the trees into a propagule and then fall down and then establish as a seedling. But if your seedlings are not surviving into the sapling state and are not rejuvenating your adult population, then there's probably something happening there. If we look at Isipingo, you have a very dense canopy, and your seedlings are very much on the edge of the channel where there's a bit more light. So those very stable adult trees are probably blocking out the light for your seedlings to establish. So this is the work of Danielle Roach, who's just finished her master's and will graduate in a few weeks. And she found even with the crab hole abundances, there wasn't a big change, or with the Ceratidia. So the mangroves in KwaZulu-Natal seemed to be quite resilient to that particular flooding event. And we can look further afield as well, because remember, our mangroves compared to the rest of the world are not on the same level. So if we look at the mangroves in Kenya, for example, they grow much faster, they grow much bigger. If we go to Asia and Australia, our mangroves are babies compared to the rest of the world. So if we have an example of hurricanes, so we know within the Florida Gulf system, there's a lot of hurricanes that come through. And that really influences the mangroves there. So how resilient are those mangroves to impacts such as floods. So if we look at leaf area index, and that's just the amount of leaf area that's present, obviously if a massive hurricane goes through, you have not just rain, but you have wind, and it's really the wind that's having a major impact. So you lose a lot of your leaves. And they find that after 1 year from a hurricane, your leaf area index recovers to what it was before the hurricane. If we look at carbon, and of course we're all very sensitive about carbon because mangroves form part of a nature-based solution to climate change. So the amount of carbon that can be lost during a hurricane can be redeemed within about 4 years within that particular system under those conditions. That's probably not the same as South Africa because we know our mangroves grow much slower. So it's important to look across the world when you're trying to understand what is the resilience of our mangroves to these kinds of changes. One of the negatives is sedimentation and high levels of sedimentation. You need sediment, mangroves need sediment in order to build land, but there's also examples where sedimentation can have a negative impact. And this is happening right now. So also from Danielle's master's project at Mgeni Estuary, you have the Beechwood Nature Reserve. And if you go to a portion of the mangroves that are just adjacent to the sand dunes, you find this very interesting phenomena where the sand is moving into the mangroves, and you can see a very clear line between the rich, dark black sediment that you would ordinarily find in your mangroves compared to this orange, sandy environment that's now present. So we looked at what is the impact of that sand. We know that it's killing the mangroves because it's essentially burying the roots. So your aerial roots are decreasing. The crab holes are completely gone. So the crabs don't like that high sediment environment, your mangrove crab species. And of course, the sediment characteristics themselves are very different. So if we look at moisture content and organic content, you have a massive drop in both of those factors in areas where deposition is taking place. And this has been driven by sea storms. So it's a very narrow beach on that side. And when you have these massive sea storms coming in, it pushes that sand down into the mangrove forest. And it changes the sediment particle size, which would change the levels of anoxia within the sediment, and it alters the way the mangroves can function. So we find that there's a lot of dead trees in that area, especially for Avicennia and Brugueria. And those two species are quite similar in terms of their root structures because the roots come out from under the ground and they're not very tall, say about less than 50 centimeters above the sediment. But for Rhizophora mucronata, which is the red mangrove, because the prop roots come out from the main stem, those trees seem to be able to withstand that sand deposition because those prop roots are not being covered as fast as Avicennia and Brugueria. So those aerial roots are very important for the way the plant functions. And of course, it would be strange for me not to talk about microplastics. So when we look at our microplastics and how they affect the quality of the habitat, we can look at it in terms of what's being consumed by the different kinds of animals. And in every animal we've analyzed so far, we've always found microplastics, and it's usually fibers that we find within them. But we also need to look at what is the contribution of microplastics to our carbon stocks. Because remember, plastic comes from oil. So it's also adding to our carbon stocks within these blue carbon habitats. So it could be a limited contribution depending on the pollution status of that particular estuary. It could also depend on which polymers are present within those systems because some polymers have more carbon than others. So we know zonation will affect microplastic distribution because our macroplastics are degrading and fragmenting into microplastics, and those are being trapped at different places within our mangroves and salt marshes. And of course, we know our plastics are also interacting with our different biota. So this represents a knowledge gap within South Africa How do we assess it? What can we do about it? So just on this side here, this is an example of the filter that goes onto our FTIR machine, which was very nicely sponsored by the JML program. And our FTIR machine is able to tell us which polymers are present within this. So you can see this looks like quite a dirty sample. There's lots of fibers, lots of fragments. Not all of those fibers are plastics. Not all of them are polymers. Some of them are going to be cotton. So we need to be able to verify our data. So this is some of the preliminary data that's come from Siyanda Butelezi's project. He's in the first year of his master's. We're working at Durban Bay Estuary or Durban Bay Harbour, where we just did a preliminary one here. We looked at 5 centimeters and 10 centimeters, how much carbon is present in those layers and how many microplastics are present, and what are the different kinds of polymers that we can find within those layers. So when we go back in January, we're going to try and do a 50 centimeter core, and we'll be able to slice that core up into different layers and analyze those layers both for carbon as well as for microplastics, because we know that there's a trend in terms of where is the carbon deposited within our different layers, and we'd like to see if that correlates to where our plastics are as well. So if we move on to the difference to salt marsh, so as we get from this part of the country and we go up west, we have large areas of salt marshes and reeds and sedges. So when I moved to the Western Cape, I knew that salt marshes was going to be very much in my future. And this is from the Berg River Estuary. How many of you have been to the Berg? One of the most beautiful estuaries in the country. And if you look at the salt marsh, it's almost like art. It's just the different colors of the different salt marsh species are quite spectacular. So if we ask the question again, how do we measure the health or our status of our salt marsh? Again, we can do it in cover. For our salt marsh species, we can look at the plant species diversity. We can't do that very well for our mangroves because there's so few mangrove species in the country. Again, we can look at carbon storage. We know for our mangroves, vegetative growth is very important, but there are also species that will regenerate from seeds. The levels of pollution, again, our sediment becomes critical because some salt marshes would prefer very dry compared to others that would prefer much wetter habitats. So this is a project by Kezia Dreyer that we're doing at the Berg River Estuary. And here we're trying to look at how do the different threats affect the amount of carbon that's stored within the sediment specifically. We know there's a number of land uses, different kinds of land uses at the Berg River Estuary. We know that there's been large modification of the mouth. The mouth is now permanently open. So there's a large amount of water coming in and flowing out, and that's led to a lot of erosion. In some places at the Berg River Estuary, we lose about 1 meter of salt marsh over time. How do they know that? Farmers have put up sticks within the habitat, and now some of those sticks are sitting out in the estuarine channel because we've lost that much of salt marsh in that particular estuary. So we know that there's the Berg River Dam, so there's a lot of abstraction, and that's changed the sediment budget within that system. And then, of course, there's pollution, there's agricultural return flow, wastewater from different land uses, and there's also illegal and overfishing happening within that system. So Kezia worked really hard on this, and we're hoping she's going to submit this year still. But she was able to go to specific species of salt marshes and to get our carbon cores from those species, which is not something we do often in the country. And then we back that up by going to areas where we know threats were taking place. And those threats were farms, different walkways that cause fragmentation, the salt pans. At one point, we thought the salt pans were decreasing in the system. Now they are increasing the amount of salt pans, as well as the impact of erosion. And what we found there, if you just focus in on the third bullet, the amount of sediment organic carbon stored within our species, so our intact species, is much higher than what we would expect, what we would find within our threats. So areas that are impacted by farming, walkways, salt pans, immediately lose a lot of the sediment that's in, a lot of the carbon that is in the sediment, and that's because that sediment characteristics change with those different impacts. And we know that our disturbed sites create a very stressful condition for our salt marsh plants. So you will have a change in species diversity because of those changes in sediment characteristics. When we look at sediment characteristics, what are we looking at? Electrical conductivity correlates quite well with sediment salinity, so the amount of salt that's present. Dry bulk density and moisture content are quite correlated to each other. And of course, sediment particle size can have an impact on all of those characteristics. So there's definitely restoration potential within the Berg system. The municipality there has been a little bit proactive about protecting the salt marsh, and that's probably because they're worried about the erosion that's there. So they've put up pillars to stop people driving when they want to go fishing on the salt marsh. There are some boardwalks that are present so that you don't have to walk on the salt marsh itself. And they are very interested in having this kind of research done so that they can improve their particular habitat. The Berg River Estuary is known for the bird species that use it, so it's a Ramsar site for the birds. And we've had the great pleasure of working with BirdLife South Africa this past year. So this is the data from our honors student, David Rispel, who looked at how can we rehabilitate and revegetate some of the banks along the Berg River Estuary using reeds and sedges. So I'll just draw your attention on that side. So that's David and Philip. And you can see behind them, that was taken in winter. The Phragmites stand doesn't look very good. It's very, very brown. The picture at the bottom was taken in spring. So after all of that fresh water came in, you had a massive increase in the amount of green biomass within those systems. You can also see the level of erosion, how much sediment has actually been lost in that environment. And in some cases, you think to yourself, well, this is really a lost cause, because even within this year, they planted in February, and they plant about 50 centimeter high blocks of Phragmites. And within a few months, all of that had washed away, or quite a bit of it had washed away. And they had to go back and replant, and they had to change their methods. So now they're using hay bales to protect against the bank and to protect the plants that they have put down. So this was quite an interesting project for us. It was one of the first times that we had looked at active restoration and how that active restoration influences the sediment carbon stocks, and how quickly does the sediment start to go back to what we would have under control conditions. So on the diagram on that side, we have our control site, which was that very green patch of reeds and sedges. And we have our 3 sites where planting took place. So we have a restoration on the far side and post-restoration. And we can see within the sites where we had plantings, after about 9 months, the amount of sediment organic matter did start to change. If we look at these diagrams at the top here, so pre-restoration, the similarity of our sediment characteristics were very different in our degraded versus our control site. But after about 9 months, they started to become much more similar. And then at the bottom here, we're looking directly at the plants, and we have to divide the plants into your below ground biomass, which is your rootstock, which is very important for regeneration. We have our green biomass as well as the brown biomass of the what we can see above the ground. And we can see that over time, before or during winter compared to after winter, that freshwater inflow really had a huge impact on the amount of green biomass within our reeds and sedges. So this is just the first year. We would have to go back almost yearly to see how successful this particular project is going to be. And if the way they are planting and the species that they are planting is suited to the conditions of the Berg River estuary. And then we go to our submerged macrophytes, and I'm glad Nikki is here because we do quite a bit of research on the seagrasses and how different fish use the seagrasses. And a little bit later on, you'll hear from our master student, Omfumetse Mokwa, on her project that's at the Crom Estuary. And for seagrass, we really need to look at the plants themselves. So we need to look at their biomass above and below ground and their leaf parameters. So we know that there's a lot of phenotypic plasticity within our seagrasses, and they respond to the changing environment. We also know they're very important for carbon. They have vegetative growth, and they support their plants. They support animals, both invertebrates that are on the leaves themselves, that are in the sediment, as well as the different fish that you would find. So I wanted to highlight this study. So this is not from my research group. This is from Sophie van der Heerden's group at Stellenbosch University. And they are really driving seagrass restoration research in the country in collaboration with Janine Adams from Nelson Mandela University. So our Zostera capensis is endangered, and we have an estimated loss of about 8.3 hectares per year, and some of that is due to the natural dynamics of our estuary, and others are due to changes in our habitat quality. And I'm sure Nikki will remember in February when we visited Crom, there were high levels of nutrients that were coming out of the marina, and that led to an increase in macroalgal biomass within the system. So that changed the habitat quality, and that would have affected our seagrass. So restoration successes have been quite variable depending on the type of restoration you do. In Katie Watson's particular paper, they compared, is it better to plant individuals or is it better to plant clumps of seagrass? What kind of arrangement is needed for those clumps and individuals, and of course your planting sites. And they found in their project, and that was at Langebaan, there was an initial decline and then an increase in the area that was covered with seagrass, and it was able to be colonized by your invertebrates. And your invertebrates can have both positive and negative impacts on your restoration. Throughout the Western Indian Ocean, seagrasses are really vital. And if you went to Wyomsa this year, we heard a lot of presentations on seagrass restoration, and there's a lot of input on restoration plans and what's the best way to do it. And they've also found that using cores or plugs of seagrass, just like Katie's presentation, creates, or you have more success using those. And that's probably because you're not disturbing that below ground biomass, which is quite critical. So there's a lot of great work coming out of Tanzania, out of Professor Blandina's lab, where they have really good successes with seagrass restoration. We mustn't forget that these habitats overlap. We have our mangroves adjacent to salt marshes in this part of the country as we go towards East London especially. We have our mangroves interacting with our seagrasses and our seagrasses interacting with our salt marshes. So we need to keep those keep those in mind. And we've done quite a bit of research, and coming to Rhodes in 2011 introduced me to a lot of people at SAIAB, and therefore I could start working with a lot of people at SAIAB, and we've kept that going, which is really great. And one of the things we've looked at is that nursery function of our mangrove seagrasses and salt marshes. And we find that our mangroves and salt marshes, yes, they're quite important for that movement into them, but they're obviously unavailable to your fish at a low tide. And the seagrasses really come out on top when we look at habitat importance. And I think this was some of the work that came out of Tim Leslie's work when he was here at Rhodes. And we looked at how are the fish using our different habitats and where do we find the greatest abundances when we compare Zostera capensis to Spartina maritima to our different sandflat areas. Other work that we've done on other animals are the crabs. So if we look at areas where mangroves are expanding into salt marshes, and we know that around the East London area to Chilumna, I think there was even an individual at Keiskamma at one point. So mangroves are definitely they were planted in areas where they didn't occur previously, and they are thriving in those particular areas. And now we're finding that the crabs that are usually associated to those mangroves are moving as well. So in this master's thesis by Chaitanya, she looked at the different crab assemblages in your mangroves versus the ecotone, where you find both habitats versus your salt marsh areas. And there were very specific assemblages within those different environments. So we need to, when we look at our habitats that are co-occurring, we need to look at connectivity, we need to look at movement, and we need to look at our nutrient flows, not just the animals and the biodiversity. How many minutes? 1 minute? Okay, I'll go very quickly. Luckily, this is the last part. So I do want to bring in what's happening on a regional scale and on a global scale. So we talked about the Western Indian Ocean Mangrove Network, and we have a network of about 9 different countries where mangroves are really prominent and very important. And there's a lot of impacts on those different mangroves. And what we're trying to do is to put together a mangrove vision as well as a mangrove action plan. So the vision is a healthy and resilient mangrove ecosystem by 2050 that benefits both people and nature. We came up with, I think it's about 7 different objectives that we want to focus on in order to achieve that vision. And 2 of those is restoration and rehabilitation. And there have been successes and negatives from the region, but we're really working on how to do that properly. And then, of course, innovative monitoring, because we need to monitor how successful those activities have been. And that's where the Global Wetlands or GLOW partnership comes in, where we're going to be using AI in order to track our crabs in terms of abundance, species richness, and activity. And hopefully the next time I come to speak to you, I can show you how we're going to do that. So to end, there are many groups around the country that are looking at that have had some successes with restoration. We have to fix the systemic issues that are impacting on our estuaries. And in those cases, we can have non-human mediated recovery if we fix some of those issues. It's important for you to know that you have a role to play in generating knowledge, and that knowledge can be used in management, restoration, improvement, but also education and passion and commitment. So it's important for you to communicate what you're finding to greater audiences and to speak to a diversity of people. So the research you do matters, and it needs to be communicated in a variety of ways. And I'm often inspired by the people at SAIAB who do that very effectively. So I'd like to acknowledge my team of postgraduate students and my collaborators, the continued support of the NRF and of course NRF-SAIAB for the JML, as well as the PUCLISA program and all our other organizations. Thank you very much.
Thank you very much, Anusha. I really appreciate it. Fascinating talk. Really enjoyed it. Just for the online speakers, feel free to put your questions in the chat section on Zoom if you want to ask any questions. We're running a little bit behind, so we've got time for 1, maybe 2 burning questions. Does anybody have questions for Anusha?
It's about the students, Francesca.
Thanks, Anusha. I just have one question. A lot of these restoration programs are relatively short term. Any lessons learned from outside other parts of the world in how do you make it work, and is restoration really effective on the long run? Thanks.
We can look at Kenya for an example there. So the Mikoko Pomoja project, which was one of the very first carbon credit projects, where the community has been explicitly linked to the conservation and the planting. So not only do you conserve the mangroves that are there, but there's continually planting happening as well. And of course, those carbon credits then bring in the finances that can be used for a variety of things. So that's one of the projects, yeah.
One last question, Albert.
Thanks, Anusha. Good to see you.
Yes.
You raised something that's very crucial at the beginning, where you said estuaries, extensions of our sewage maturation ponds. And throughout your talk, you mentioned a number of estuaries from the West Coast, the South Coast, East Coast that are heavily impacted. And then there are all these ongoing efforts for restoration. But the sources of the impacts are coming from the catchments and also the municipalities. We can do everything that we want, that we can within the estuaries themselves. But if we don't address the sources of the impacts, we will not win the battle. So my question is, how effective are we engaging with all those different partners to really have effective impact? Thank you.
Thanks. So I can draw on maybe the estuarine forum groups that we have, especially within the Western Cape, where a particular estuary has all the stakeholders that come together. And through those forums, you have the development of your catchment management plans, so that you're not just working on what you can see down at the bottom, but you're also working at further up. So I know the Berg does that quite well. I think the Great Bruck is starting to bring that in. But yeah, it's fixing those systemic issues and making sure that the right people are in the room, so you are inclusive and not exclusive in your networks.
Thank you very much. I think that's all we got time for. Thank you very much. So we're just going to dive right into the next session. Just to let you guys know, for the speakers, we will hold up a yellow sign when you've got 2 minutes left to speak and a red one when you've got 1 minute. So look in Sipasiso's direction if you're wanting to see if you're going to run out of time.
Good morning, everyone. So I'll be chairing the first session. So I would like to welcome Tembani, who will be presenting on linking the juvenile bursaries to adult stocks in spadids using natural tags.
Morning, everyone. My name is Tembani, and I'll be presenting one of my PhD chapters, and it's on linking juvenile nurseries to adult stocks in sparid fishes using natural tags of multiple tissues.
Thank you.
I'm not sure why it's not. Not sure why it's not sharing the Zoom. Gosh.
So just to give some background, coastal habitats are one of the most important ecosystems as they provide a number of ecosystem services. And one of those is the one I'm going to be focusing the most, which is provision of nursery for juvenile fish and also for invertebrates. So those habitats are usually correlated with having high densities of juvenile fish. And that is usually because they provide protection and food resources for those juvenile fish during their nursery. And also, on top of those requirements for a habitat to be considered a nursery, there is a new requirement, or a recently new requirement that was added, which is the connectivity between nursery habitats and adult habitats. So in that one, they look at how much a habitat is contributing to the adult stock. However, measuring or quantifying that movement, the ontogenetic movement from nursery to adult habitats, can be a bit challenging. And there is some methods that are used or techniques that are used to try and measure that. And those range from comparing the community structure of the fish, if there is any similarities or differences in size structure, which may infer that the fish have moved from certain habitats to other habitats. And then that can also include artificial tagging, which includes your external tags and your acoustic tagging. But the thing is, some of these techniques are labor intensive and expensive. And there's been a growing interest on techniques such as natural tagging. So basically with natural taggings, the fish gets the tags based on the environment that they've been occupying the most. So those are your stable isotopes, which are usually associated with what the fish feeds on, and some of them can be associated with the physicochemical parameters. Then you've got your trace elements or your microchemistry, which is usually showing the surrounding environment of the fish. And also, if you study them on the right tissue, then you can be able to trace the entire life history, habitat use of the fish. And usually those tissues are archival tissues such as your otoliths and your eye lenses. So for this study, we aiming to use these natural tags to determine habitat usage by juvenile sparids in San Francisco seascape, and to further quantify habitat connectivity. So we'll do that. We did that through the following objectives, which is to use the natural tags to assess how these sparids utilize the studied seascapes during their nursery life stage, to create the trace element baseline for juvenile sparids collected from different nursery habitats within the seascape, and then to quantify connectivity between nursery and adult or spawning habitats. So this study was conducted in San Francisco, as I've said, and we collected our fish from Hamtus estuary and Chrome estuary. And we also collected some adults from the nearshore marine environment, but I'll be focusing on the estuarine ones. So the main differences between the two estuaries are the extensive seagrass at Chrome estuary, and around Hamtus there is also some farming that is happening there, which may come with pollution. And also Chrome is also known as the arm of the sea, so it's practically mostly marine dominated. In terms of species, we're focusing on the Cape stump nose and the stripey. The Cape stump nose is a marine estuarine dependent species that is usually associated with seagrass in estuaries when they are still at nursery stage. It is endemic in South African waters, and the stripey, on the other end, is a herbivorous species. It is a marine estuarine opportunist, which means it does use estuaries and the nearshore marine environment at nursery stage. It is mainly found in Southern African waters and in the Med. We looked at stable isotopes, the carbon-13 and nitrogen-15 stable isotopes, to look at the recent utilization of nursery habitats by the fish that we collected. So basically we removed muscle tissue, make sure that there's no scales attached to the tissue that we're gonna use, no scales, no bones. We freeze dry it over 24 hours, grind it to powder, and then we pack it in tin capsules and send it to BioGrip in Stellenbosch for analysis. And then for trace elements, we remove the otolith and we polish it to the core. And then we mounted them on slides for laser ablation. And then the laser ablation was run in Lund University in Sweden. And the laser ablation was run from the edge of the otolith through the core to the other edge of the otolith. And we quantified about 14 elements, but I'm only going to be focusing on barium, strontium, manganese, and magnesium. Barium is a brackish water proxy. It goes higher in brackish waters and it is lower in proper, stable, high salinity areas. And then strontium, on the other hand, it is a salinity and temperature proxy. It gets higher in high salinities with stably high or low temperatures. This graph here is just showing you the relationship between some of the archival tissues and the fish growth. Like I said, those archival tissues are important when you're using natural tags. That's because they grow as the fish grow. So you can see your otolith on the left and the islands, they all have a strong positive relationship with total length. In terms of stable isotopes, when we look at on your left there, we've got an isoscape that is showing the feeding ecology of both the Cape stump nose and the stripey. So if you look in the absence of seagrass, the Cape stump nose is shown there by, is that blue or green? I think it's green. It's shown there by green at the top. In the absence of seagrass, they feed on the higher trophic level compared to the Cape stump nose, which is shown by orange, that is collected from the system with seagrass. So we're assuming that in the absence of seagrass, they feed on things like your polychaetes, but in seagrass, they are known to feed on epiphytes and epifauna that is associated with seagrass. Hence, they feed on the lower trophic level. And then the stripey, on the other hand, which is our herbivora species, feeds on the lowest trophic level. It's there indicated by gray. And for it, it's probably feeding on epiphytes also, but and also feeding on seagrass itself. And then the ones that are collected from chrome, they both have a bigger niche size, which speaks to more food resources available for them. And then the graphs on the right are just showing the differences between carbon-13, how different carbon-13 is when you compare the two groups, which is basically the same thing I just explained on the isoscape. And then when it comes to your trace elements, BA for species that are collected from chrome was much higher and significantly higher compared to the Cape stump nose and stripey collected from the barium that is for species that is collected from hamtooth was much higher compared to the Cape stump nose and the stripey that is collected from chrome. And then manganese to magnesium ratio followed the same suit. And then when it comes to strontium, there was no significant difference. And then what does this mean when we look at single fish profiles? So basically the Cape stump nose, especially the one collected from hamtooth, both Cape stump nose collected from hamtooth and the one collected from chrome, they spawn at sea. You can see here, I'm not sure if we have a pointer. Do we have it? Okay, you can see on the left, at the top, I've got the strontium graphs, and then at the bottom, I've got the barium graphs. Then top left, I've got the Cape stump nose collected from hamtooth, and in the x-axis, the This is the distance of the otolith, and the 0 is representing the core. This is where the life started for the fish. And you can see strontium is higher there, which means the spawn at sea. And then barium on the lower graph starts at 0, which means they spawn, which also confirms that they spawn at sea. And then we've got a region that is represented by the black arrow, which is when they recruit into estuaries, shown by the dip in strontium and an increase in barium. And then from there, they just utilize the entire estuary. And then you can see the last arrow there, which is orange. That is where they going down in the lower reaches and preparing to leave the estuary. And then here for the opportunist, it is quite a different story for them. Both strontium and barium don't show any proper trend, but we managed to use the manganese per magnesium ratio, which is a hypoxia, which is a hypoxia and wetland proxy. So we assume that where it started going up, it's when it started interacting with the water that is coming with pollution in estuaries. So we may say that that is when they entered estuaries, and they seem to be staying in an estuary there because it's fluctuating in the same or in a certain range. This is an ongoing project. So from here, what we're planning to do is to analyze the otoliths of the adults and then see if we can pick up the juvenile phase of those adults and compare to the baseline that we have created. And then we see if there is any differences. And then we try to quantify where they actually come from in these two estuaries. And then we're going to repeat the same process on islands and see if they're going to give us any different results, because these are two different tissues, with the otolith having or being made up of the calcium carbonate, which is a poor form, and then you've got the islands, which is made up of protein, which we assume is going to give us some better results for some elements. And then we're exploring stable isotopes on islands, is where we peel them off one by one and we analyze them and we see if there is any differences that can show where actually they were feeding when they were moving up and down the estuary. Thank you so much.
Thank you very much. We've got time for just one question. If there are any questions. No questions? Okay, then thank you very, very much, Timbani. Appreciate the talk, was very interesting. The next speaker is going to be Samkele, and he's going to be talking about ecological niche modeling and thermal performance to quantify the climate change response of diplodes. And this will be a speed talk.
Good morning, everyone. So I will be telling you about the findings of my master's thesis. So we did a species distribution model to model the contemporary distribution of Diplodocus capensis along the South African coast, and we found the species to be quite widespread, occurring in all four major biogeographic zones. However, when we looked at the temperature and what it's been doing over the past 40 years, we saw some cooling trends and warming trends, so different temperature trends along the coast, which then allowed us to then look at the future distribution of the species. And what we actually noticed is that on the on the west coast or the western edge of the species range, the species is likely to lose about 22% of its distributional range. So when we went back and looked again, we found that while this is driven by cooling trends on the west coast, we're seeing cooling trends in the middle of the core range, but we're not seeing any changes in the distribution between now and the future. So we then asked the question as to why this is happening. So this is where the physiology comes in after the species distribution modeling. So with the physiology, what we did is we went out and we caught 20 adult blacktail, and we subjected them to intermittent flow respirometry, where we measured standard metabolic rate and the maximum metabolic rate. This then allowed us to calculate what is called an aerobic scope for the species. And what we found for the aerobic scope for blacktail in that area, in the core range of the species, is that it's got a biphasic relationship with temperature, meaning that it peaks at around 16.89 degrees, and with suboptimal temperatures at both 13.74 and 20.05 degrees, meaning that anything below 13.74 and anything above 20.05 degrees is suboptimal for the species. What this allowed us to do was to take a retrospective look at what's been going on, in the species core range over the last 40 years. And what we actually saw is that very agreeing with the cooling trends, so that the number of cold days has been increasing over the last 40 years, while concurrently the number of warm days has been decreasing for the last 40 years, meaning that the suboptimal part of the suboptimal end, or the cold suboptimal end of the Page's temperature is now becoming more of an issue for the species. What then we were able to show is that you can sort of use species distribution modeling along with physiology to answer questions that species distribution modeling can't really, address. So, for instance, it will be able to detect changes in the edge of the range, but it won't be able to detect changes in, the middle of the species range, which then where physiology comes in. So we were able to show this with this project. And what another big finding that we had here was that with the loss of the species range in the West Coast specifically, being predicted to occur sometime at the end of the century, there is a real risk to the small-scale fishery that is occurring, that is currently taking place there, which could lead to a loss of livelihoods and potentially a shift in fishing effort to other more cold water tolerant species, which then begs the question for, it begs management questions for that area, which we will then need to answer in the future. Thank you very much.
Thank you very much. That was spot on, down to the second. Do we have got time for some questions? Any questions? The mic is coming. Run, run, run.
Thank you for your presentation. When it comes to modeling, one of the most important things is variables. I want to ask you about your variables and how many are they, and what are the more important ones. If I miss, sorry. Thank you for.
Okay. Yeah. Thanks for the question, Amir. So we used 4 main variables to begin with. So we had bathymetry along the coast. We then had 3 sea surface temperature variables. So we had summer sea surface temperature, winter sea surface temperature, and annual sea surface temperature. When we did the model selection, we found that, annual sea surface temperature didn't matter as much. So we then did a reduced model that had summer, winter, and bathymetry. And when we looked at the variable importance in that, or the variable contribution, we found that summer is the most, important sea surface temperature variable. And when you look at the actual summer trends, you can see that what's happening where the species ranges are changing, it's when you see strong cooling or strong warming. So, I mean, it sort of demonstrates how unique the South African coast is, because mostly around the world, it's mostly like warming trends. But here we see a mixture, and that mixture is affecting fish in different ways, but it's causing the same risk, So those are my variables. I hope you answered. Yeah. Sure.
Thank you very much. Rehan. Okay, our next speaker is going to be Kevin, and it's an online talk, so it's going to be pre-recorded. Just give us a second while we just set it going.
This one is max out.
Yeah, sound. Sorry, we just got a technical issue here. Is it playing? Okay, because we've got a bit of a technical issue, I think we're just going to switch around the talk. So, Apiwe, would you mind come to give your talk now, and then we will come back to Kevin after that. So we're just doing a switcheroo. So I didn't introduce you. Apiwe is going to be talking about seagrass estuaries and the effects on coastal habitats.
Just left or right click in terms of change it. Okay. Left arm. Perfect. My name is Apiwe. I'll be sharing my master's project whereby I looked at the nursery quality of different coastal habitats based on juvenile Cape snubnose condition and diet. Coastal habitats differ in their structure. Coastal habitats differ in their structure and productivity, and these differences shape how these habitats function ecologically. For example, vegetated estuaries offer juveniles good resource provision in terms of food and shelter from in terms of predators. So in the diagram that you see here is a Cape snubnose, whereby they spawn in the nearshore environment, whereby their post-flexion larvae juveniles recruit into these estuarine habitats, and they have enough resources in terms of food and shelter, and thus connect to adult habitats. But more variable coastal habitats might not offer the same resources. So an area whereby juveniles occur, we can't just classify as a nursery habitat. So where juveniles occur, we'll consider that as a juvenile habitat, and whereby if that coastal habitat offers enough resources in terms of shelter and food, then we'll consider it as a nursery habitat. And building on the general idea of nursery habitats, Beck first formalized the nursery habitat framework in terms of looking at fish species that use different habitats in terms of the juvenile and adult phases, and then also looking and comparing the juvenile habitats that and also comparing the juvenile habitats that these species might occur in. So basically, you can't just look at, but you can't just identify a nursery habitat by looking at where the juveniles occur. It involves looking at the structural complexity of that area, temporal variations, and resource provision and how that might influence how they grow. And the main four factors was habitat connectivity, resource provision to support higher growth and survival, and moving and connecting and moving towards adult habitats. So this study by James provided a foundational framework for my study. Most of the studies in the past, they just looked at juvenile abundance, and that gave them an idea that that was a nursery habitat, but you had to look at structural complexity, feeding ecology, and connectivity. Also, to provide some background, one of my sites I'll be talking about are stromatolite pools. stromatoli pool. This was the first study that looked at how fish communities interacted with stromatoli pools. And one of the fish found within these stromatoli pools were the Cape stump nodes and some freshwater mullets. But the idea was whether or not these stromatoli pools supported the Cape stump nodes in terms of providing enough food and resources, and whether or not they were acting as ecological traps. There are certain traits about these stromatoli pools that result in the juveniles recruiting in them, which I'll expand on in the next few slides. I'm not a geologist, but I think I'll be clear about this. Stromatoli pools have two depositional models. One of them is the pool system, which is the one in this diagram. So freshwater seepage mixes with wave intrusion at high tide and through wave splash, which mixes with the fresh groundwater, which results in salinity gradients within the stromatoli pools. Mineral precipitation forms these rimstone dams, which have stick-like pools that occur all year round. And another important trait of these barrage pool systems is the macrophytes associated with them, which mostly occur in wetlands and in estuarine salt marsh vegetation. So my thinking is that due to the fresh water coming into these stromatoli pools, this results in the juveniles, like the Cape stump nose, recruiting into the stromatoli pools because of the olfactory cues. And the important thing for me to do is looking at whether these stromatoli pools support juvenile growth and survival. So the aim of my study was to look at the necessary quality of the juvenile habitats by looking at the Cape stump nose condition, morphology, and feeding ecology. I had 3 sites, which was Seaview, Gamtoos, and Chrome. Seaview was the stromatolite pool, the barrage pool system. Gamtoos, which we've categorized as the non-seagrass estuary, and Chrome as the seagrass estuary. So for my methods, I looked at condition, and first of all was Fulton's condition factor, which was basically calculating length-weight relationships, gives you an idea of nutrition, what they're feeding on, how well-fed they are in terms of the length-weight relationship. And then I used the CNS elemental analyzer by looking at the fish, muscle, tissue, carbon, and nitrogen ratios, which will give me an idea of condition as well. A high carbon to nitrogen ratio will give me an idea that they have high fat storage in terms of lipid ratio, and a low carbon to nitrogen ratio will give me a low lipid ratio in terms of fat. And then I'll be looking at otolith shapes. This will give me an idea of their growth rates and how the habitats that they're in might be affecting them in terms of environmental stress. And then I looked at morphology, which is also looking at the body shape, whether they have deeper bodies or streamlined bodies, whether there's any morphological plasticity due to the habitats that they occur in. And lastly, I looked at feeding ecology. That involved looking at gut content, which is a short-term way of looking at feeding ecology, and stable isotopes, which is much more long-term, looking at carbon-13 and nitrogen-15. So for my results, as you can see in the middle, the Chrome site, which is the seagrass estuary, had the higher condition factor, about 1.87, with Gamtoos, the non-seagrass, and Seaview, the stromatolite pool, at around 1.56, both of them. And then I looked at morphology using TPS-DIG, and then you can see that the juveniles caught at Chrome had deeper bodies, which is associated to a vegetated habitat in terms of structural complexity. And then Gamtoos and Seaview were more of the same streamlined bodies related to an open area. Basically, this is a CVA. We looked at it's basically the same thing, but in terms of the geometric morphometrics, differentiating them in terms of their shapes. So this is my stable isotope results. As you can see, the ones at Seaview had a wider isotopic niche. Basically, the ellipsis gives you an idea of their dietary variability. So Seaview had a wider isotopic niche, so telling me that they have a more generalized diet as compared to Gamtoos, which which is smaller, so maybe a specialized diet. And I'll expand on that later in terms of relating it to my gut content. And chrome, which was more medium comparison to the other two. The interesting thing here about the box plots, still showing the isotope data, is the Seaview site. They had the highest nitrogen 15 variables. So when you look at nitrogen 15, you want to look at whether they're feeding on a higher trophic level. Basically, I haven't collected my baseline data yet, so I can't assume they're feeding at a higher trophic level because you have to understand whether it's a food web effect or a nutrient effect. So I'm thinking in terms of the fresh groundwater coming into the stromatolite pools, that might result in higher nitrogen levels. This is my gut content. I finished it about 4 weeks ago, so I haven't actually looked at the data in itself, but I still wanted to talk about what I've seen so far. At Chrome, most of the juveniles in that area had full stomachs with a lot of seagrass and invertebrates related to the seagrass, showing that they have high prey availability. At Khamtos, there was a lot, it was mixed. Some of the stomachs were empty, some were full, and they were mostly feeding on invertebrates with a lot of gastropod shells. The Seaview guys, shame, they're struggling. Most of them had empty stomachs, but I did find a couple of amphipods in their stomachs. And I also have this theory running because I also found gills within the gut content. So I also think that they might be feeding on shrimp that are found within the stromatolite pools. So basically, my ideas going into next year, and hopefully my results, when I do more of my methods and get more results, I have concrete ideas on this. Chrome is functioning as a high quality nursery habitat, offering good food, which you saw with the high nutritional condition in terms of Fulton's condition factor, and also with the gut content, the amount of full stomachs that were in there. Then the non-seagrass estuary Khamtos as a juvenile habitat, not a nursery habitat, but do some sort of way support the juvenile habitats there, the cave stump nose. And I think the Seaview stromatolite pool is functioning as a stepping stone habitat in terms of connecting to other estuary habitats. They move into there, shortening the pelagic larval duration stage, and then getting at least enough amount of food, and then moving elsewhere because it's a struggle there, as you can see. Thank you.
Thank you very much. So we do have time for questions. You've also finished spot on. Anybody with any questions? There we go, at the back.
Hi, up here. I actually did a similar project for my honors on freshwater mullet. And I found that with the mullet, there were no differences in the condition. I mean, in the ones that we sampled, between stromatolites and estuary, we're working on the Swartkops. I'm not sure if I missed it in your talk. Did you find any big differences in the condition? And maybe are you finding that the ones in the because what we found was crucially is that the ones in the stromatolite pools, while the quality of the food is not great, they are more generalist. So they'll eat anything, they're more opportunist. Did you find that, or are you still?
In terms of the Fulton's condition factor, the nutritional condition box plot that I showed there, at Chrome, they had a higher condition factor index, showing that they were fatter, basically. And then the ones at Khamtos and Seaview, which Khamtos is the non-seagrass, basically the same.
Okay, Khamtos and Seaview are similar.
Yeah.
Okay.
Perfect.
Cool. And the niche space, is it bigger?
With at Seaview, as you said, they're generalists. They had a wide isotopic niche, so feeding on basically anything they can find. So basically, survival of the fittest, hunter-gatherers. Anything I can find, I eat.
Thank you very much. Okay, we're just going to do a quick switch around again. We're going to have a speed talk by Melissa. Is she here? Yes, there we go. Thank you very much. Melissa's going to be talking about shallow water seascape connectivity.
Good morning, everyone. Like Gwyneth said, I'll be talking about shallow water seascape connectivity, focusing on vegetated habitat utilization by two common estuarine-associated fish species in the estuary ocean ecotone. So identifying fish nursery habitats include identifying critical settlement habitats, identifying habitat connectivity through dial, tidal, and ontogenetic habitat shifts, identifying hotspots of animal abundances and productivity in the seascape mosaic, and finding highways connecting nurseries to adult populations through ecosystem corridors. Jumping straight into it, the aim of this first study was to assess the abundance, size, class, and residency of Auraluba and decapensis to eelgrass beds in the lower reaches of the Swartkops estuary, with objectives being to determine, are these two species using the Swartkops estuary as a nursery area? Where are the core nursery areas, and how resident are these two species to seagrass beds in the lower reaches of the Swartkops estuary? For the residency of Auraluba and decapensis to eelgrass beds in Tippers Creek, mark recapture sampling consisted of 848 Auraluba and 268 decapensis being tagged. A total of 144 seinets were sampled. During the sampling period, 8 decapensis were recaptured, producing a recapture rate of 2.99%. And 73 Auraluba were recaptured, producing a recapture rate of 8.61%. This study again highlights the importance of eelgrass beds in the Swartkops estuary for juvenile Auraluba and decapensis. The loss of eelgrass beds in the Swartkops estuary due to anthropogenic impacts and pollution could cause a decrease in both species. Maintaining the health of the Swartkops system is thus imperative if it is to remain a nursery area for vegetation-associated fish species. The next study aimed to assess the residency and home range of juvenile decapensis in a shallow coastal cove known as Flat Rocks using acoustic telemetry, with the objectives being determining how juveniles are using an inshore nursery area and their residency to it, as well as determining the home range of the species within the nursery area. 19 decapensis were tagged and monitored from August to December 2021 during the monitoring period, with 11 young of the year and 8 juveniles being tagged. Over 500,000 detections were recorded during the monitoring period within the Flat Rocks array. Overall, the residency index of fish were high to the cove with an average of 80%. There was a notable difference in residency index between habitats, with high profile reef and mosaic reef having the highest residency index, followed by flat reef and deep reef 1, and deep reef 2 having the lowest residency index. The Plotus capensis is an important species for subsistence and recreational fishes and contributes a large component of the average catch. The current study showed that the juvenile decapensis displayed a high site fidelity to the shallow rocky cove known as Flat Rocks. Residency indexes were seen to be highest for high-profile reef and low-profile reef, showing that these fish prefer the food and shelter provided by the macroalgae growing in the shallow cove. The home ranges of decapensis were also seen to be small and be concentrated within the cove. This highlights the importance of shallow coastal coves such as Flat Rocks as a nursery and transition zone for species such as decapensis, and that the conservation of these areas are of importance as it supports all cycles of this species. Thank you.
Thank you very much. We've got time for one question while we switch over to the online talks. Any questions for Melissa? Just check online. Thanks very much, Melissa. Really appreciate your talk. And we've got IT now just to help us get the sort out the glitch. You know, there's always a few glitches in any presentation. So we're just sorting out the audio glitch for the pre-recorded talks. Give us 5 seconds. Thank you very much, Kevin, for your great presentation. The floor is open for questions for Kevin. Kevin, you should be online. If you can unmute yourself, Kevin, to answer any questions. Any Sorry. Any questions for Kevin? Okay. Yes, right at the back there. Yeah, that we believe the online people have got issues hearing the talk. They can see the images, but they can't hear it. We are working on it. Okay, thanks. Everyone just give us 5 seconds. I think these trials and errors come to test us. So just give us 5 seconds. Thanks.
We can
Yeah.
try.
Very much for that great talk. Do we have any questions? Mfumetsi? No questions? We've got time. We're doing very well for time, so thank you very much for everyone for sticking to time. No questions. Okay, great. So we're going to have our last talk for this session, and that's by Jessica. And Thank you very, very much, Jessica. That was a great talk. Do we have any questions? No questions? A break for tea. Please note that we're going to start promptly at 11 because we have to dovetail with the DZE, which are presenting an Eden Grove Red. So we need to synchronize quite tightly. So please be seated by 11 o'clock, but enjoy tea.